Thanks for coming along to our first webinar for Restore and Renew, which forms part of our larger climate readiness program that we're delivering throughout the Hunter and the Mid-Coast over the next 12 months. My name's Josephine O'Brien and I'm the Regional Landcare Coordinator for Hunter Region Landcare. I'd just like to firstly acknowledge the First Nations people from across the regions of the Mid-Coast and the Hunter and pay my respects to the elders past and present and recognise their long and enduring connection to the wonderful country that we honour, respect and care for. There's just a couple of housekeeping requests that we've got. Can you please mute yourself unless you're asking a question at the Q&A time? Feel free to, if you've got any questions throughout the webinar, just type them in the chat group and we'll be monitoring that and make sure they all get answered. If you do have a question, just raise your hand when we get to the Q&A time. I'd just like to introduce Tricia Hogbin, who's our facilitator for today. Tricia's the Restoration Genomics Outreach Coordinator at the Royal Botanic Gardens in Sydney. She has a PhD in evolutionary ecology, a research background in threatened flora ecology and conservation genetics, as well as experience coordinating recovery projects. And she's also president of our very local Broke-Bulga Landcare group in the Hunter. So I'll hand over to Tricia now. Thank you.
Thanks, Jo. I'm just going to share my screen and hopefully you can see a slide there now. Okay, so I'm going to be talking to you about using the Restore and Renew web tool to guide the sourcing of plants for your resilient climate-ready restoration. There'll be a lot of information presented in these slides. You'll not only have access to the recording, but I'll also give you a PDF of the slides, so don't feel like you need to take notes. So I'm with the Research Centre for Ecosystem Resilience at the Botanic Gardens of Sydney, and so we're a team of 20 multidisciplinary researchers and we all have a common interest in guiding the conservation of resilient ecosystems, hence the name, through large-scale genomic projects. Now, to give you an idea of the scale of the projects the group's working on, us and our collaborators have collected leaf samples from more than 109,000 plants across Eastern Australia for more than 2,000 species. And we've so far sequenced the DNA of 62,000 of those samples. And that all contributes to our genomic knowledge infrastructure, which is effectively one whopping big data set that has all sorts of applications and can answer all sorts of wonderful questions now and into the future. So one of those applications is the Restore and Renew web tool, which is what we're talking about today, which is genetically informed seed sourcing guidelines for 100 species so far. And over time, we hope that we have guidelines there for many hundreds of species. So today's presentation is really all about helping you or highlighting how you can use Restore and Renew to restore resilient climate-ready ecosystems. So I'll start off by looking at reminding ourselves what is a resilient climate-ready ecosystem and why the genetic diversity of single species is relevant. I'll highlight why it's time to move on from outdated generalizations regarding local provenance to evidence-based genetic neighborhoods, and how it's time to shift the focus from just putting plants in the ground to restoring plant populations. I'll introduce you to the Restore and Renew web tool and what it does, and then we'll have a web tool demonstration. I've just popped these two pictures in here to remind me to kind of highlight what I hope you walk away with today. So here we have a really healthy, happy-looking restoration of river red gum in the Upper Hunter, and look successful. Those plants are 14, 17 years old now. But what I want you to understand today is that the success of that restoration is not just about the short term, it's about the long term and whether or not those plants can produce seed and offspring that can survive and adapt in a changing climate and in the face of a whole heap of other threats that we can't even anticipate. And that all comes down to the plants or your plant selection and the source of those plants. So let's jump back. What's an ecosystem? It's a community of living, biotic things interacting with each other and their non-living abiotic environment. It's made up of populations of different species. So these populations are the building blocks of ecosystems. So, for example, we've got the bull oak or Alocasuarina lumenii here, a group of bull oaks growing together in the same place, interbreeding freely and producing offspring that replace those that are lost or those that senesce, a population. We also have this bipinnate acacia. I'm not even going to attempt to identify from a photo. They're all interbreeding and producing offspring that replace those that senesce. And likewise, we have this uke, all interbreeding and producing offspring that need to be able to adapt to a changing environment if this ecosystem is going to persist. So to restore an ecosystem, the populations of species within it need to be able to survive, reproduce, and maintain themselves. In other words, they need to be self-sustaining. So for your restoration to be successful, it needs to be self-sustaining, and to do that, it needs to be made up of functioning and resilient populations of its component species. Unfortunately, a lot of restoration to date has focused on the quality, the quantity of plants, the number of plants that are going into the ground, rather than the quality. But quality is more important if we're creating self-sustaining populations, and if we're doing something more than gardening. Where someone needs to come along and plant more plants in the future to replace those that you've planted. So for a population to be self-sustaining, it needs to be fecund, reproducing now and into the longer term. It needs to be adapted to current conditions and also adaptable to future challenges. It needs to have the capacity to adapt and reproduce under changing environmental conditions. So for a population to achieve all these things, it needs genetic diversity, non-negotiable, because genetic diversity maximizes adaptive potential and minimizes inbreeding. So in other words, where you source your plants from or your seed matters, because if planted or your natural populations in your fragmented landscapes have low genetic diversity, they may not reproduce, and they're unlikely to have the potential to adapt, and you're simply gardening. Because the more genetic diversity a population has, the more likely it is that just by chance alone, that at least some individuals have a trait that enable it to adapt to change. So we know that if we have populations with low genetic diversity, they have a really low likelihood of at least having some individuals that can adapt to a change or to a threat or to a pressure, be it climate change. Here we have phytophthora, leaf curl disease, myrtle rust, and there's an unlimited number of threats we perhaps can't even think about now. So if you've got low genetic diversity, populations are highly vulnerable to these threats, to climate change and so on. Whereas if you have a population with really high genetic diversity, the chance that at least some individuals will have the genetic makeup that enable it to adapt to a change or a pressure is higher, and they're less vulnerable. Now, genetic diversity is also important because inbreeding reduces fitness. Now, inbreeding is when individuals that are genetically closely related or similar mate and they produce offspring. So, for example, seed from the same tree or from a small population. Now, unfortunately, current restoration practices encourage inbreeding in restored populations. It's super easy to get thousands of seed from a single eucalypt branch. And when they're planted out next to each other in a restoration site or near each other, they're all siblings or half siblings. And so we're encouraging inbreeding. So let's dive into a genetics refresher from your high school biology days to look at why that might be a problem. Again, there's a lot of information on this slide, but you'll get a PDF of this, and I just want to highlight the key points. So a bit of a background. So alleles, alternative versions of a gene, and at a given location in the genetic code, each individual has two, one from each parent, and then they can be the same homozygous or different heterozygous. So the dominant are expressed no matter what, and the recessive are only expressed if not masked by a dominant allele. Now, in large genetically diverse populations, these deleterious recessives are rarely presented or rarely homozygous, so they're rarely expressed and they stay hidden. Whereas when you have low genetic diversity or increased relatedness, you get an increase in homozygosity and an increase in the expression of these deleterious recessive alleles that are normally hidden and not selected against, which can lead to inbreeding depression, which is not something you want in your restored populations because it can lead to a failure to set seed or weak seedlings, and so not ideal. So in summary, to restore resilient climate-ready ecosystems, you should be striving to restore self-sustaining populations for each of your target species. And you want to capture diversity within species to facilitate adaptive potential or resilience. And you want to strive for diversity within individuals to avoid inbreeding depression. Now, the problem is, unfortunately, material used in restoration typically has low genetic diversity due to habitat loss and narrow seed collection. So here we have a schematic representation of a happy, healthy, genetically diverse natural population. We lose some of those individuals due to habitat loss, and we know just by chance when we lose individuals in a population, we lose genetic diversity. Someone comes along and they amplify that low genetic diversity by collecting from too few individuals, which means that we have a relatively genetically depauperate restoration site, which can mean we have high levels of inbreeding and no recruitment and an unsuccessful restoration. So you can come along and someone can do slightly better. They've sampled seed from a few more individuals. We have a restoration that looks a little bit better in the short term. We've got new recruits coming along. But if we get a new stress like climate change or disease, because of the low genetic diversity, just by chance alone, we have a low adaptive potential. So ideally, what we want to do, even in our fragmented landscapes where populations are now isolated, we want to facilitate collection of a genetically diverse seed collection, which results in a genetically diverse restored population where we can see normal recruitment because we don't have inbreeding. And then what we have because of that higher genetic diversity, in the longer term, we have increased adaptive potential, an increased likelihood that some of the individuals in that population have the genetic makeup or trait that enables them to adapt. So this is probably a good time to pause and have a look at what local provenance is and why you've been told to use it. So if you could put into the chat, maybe one sentence, brief sentence, what local provenance is and why you think we've been told to use it. So yep, plants that occur in the local area, thank you. Better adapted to local conditions, exactly. I think that's, whilst you continue putting those in, I'll keep going. Yep, perfect. Here's another more detailed one. Collection of seed material within 10 kilometres of a restoration site to maintain ecosystems' health and resilience. So thank you. I'm going to move and keep going. So local provenance is effectively a generalisation aimed, as some of you mentioned, at capturing material adapted to local conditions. But really, how stable are local conditions, particularly given climate change? And also, one of the reasons there's been a preference for local provenance is the avoiding unintentional consequences of crossing individuals that are too dissimilar. So I want to briefly talk about, the consequences of crossing individuals that are too dissimilar are effectively or largely unpredictable, but they can cause a breakup of co-adapted gene complexes. So you imagine healthy, happy, large population, you have these genes, they're co-adapted, they confer some sort of fitness advantage. Everyone in the population has them because they've been selected for. But then you have an individual that is from another distant, unrelated population. And when you cross with these individuals, you can break apart those co-adapted gene complexes because the genetic code is so different. So local provenance is also about avoiding those unintentional consequences. But really, how far is too far? So one of you mentioned here, collection of seed material within 10 kilometres. I've asked this question at workshops, and someone will say 4 kilometres, someone will say 40, someone will say local government area, someone will say catchment. What the genetic data is starting to tell us is we actually can't generalize about how far is too far. So to give you an example of this, QR code there if you want to look at the publication. This is from the RESA team. We have a number of acacias and we have a restoration site west of Sydney. These grey areas are a genetic neighbourhood, effectively a local provenance, but based on genetic data rather than generalizations. So what we see here is Acacia suaveolens and Acacia ulicifolia both have very relatively small genetic collection areas or genetic neighbourhoods, whereas linifolia, longifolia and terminalis have relatively large genetic neighbourhoods. So we can't actually generalize about how far is too far, and we actually don't need to these days because genetic data is getting more accessible where we can actually understand what a local provenance is based on data rather than generalizations. It's also called genetic collection area, genetic neighborhood, or seed transfer zone. So what they are is an area across which you can safely source material to maximize genetic diversity and avoid those unintentional consequences of crossing individuals that are too dissimilar. So it's an area across which you can mix material to maximize genetic diversity. And this is what Restore and Renew provides. And so you can see a screenshot here of Restore and Renew. You can find it at this address or this QR code, but a really easy way to find it is to simply go to the Adapt New South Wales website, click on resources, and in the resources tab, among things like the NARCLIM guidance, the interactive projections map, all these useful things, is the Restore and Renew web tool. Now, for the next few slides, I'm going to use the Restore and Renew web tool for a Bulwarra. We didn't have anyone from Bulwarra Landcare here with us today, but I'm going to use it for a restoration site for restoring floodplain dry rainforest, the Rainforest Revival Project some of you may be involved in. So this is a plant community type where we have less than 3% of the original extent, and it remains as small, degraded, isolated remnants. So we've already lost genetic diversity due to random genetic drift, bottlenecks, and inbreeding. So this highlights where your restoration can help these isolated, fragmented populations persist, and you can help them adapt in a changing climate. To give you an idea, here's a screenshot of the pre-clearing extent of this lower hunter riparian rainforest. And this is what we have now. Actually, I should have said it's the red. So the red, the floodplain dry rainforest, pre-clearing, and now. So you can imagine what the consequences are if you're sourcing local provenance material from within 10 kilometres of your restoration site. You're sourcing very... you have access to a really low genetic diversity, and you're likely to be planting lots of individuals that are very closely related, and they're going to have a low adaptive potential, and they're going to be highly inbred. So this is what Restore and Renew can help you to avoid. So what it does, it synthesizes or brings together the latest genetic and climate science data to help you design evidence-based, resilient, climate-ready restoration. So behind this user-friendly interface is DNA data from more than 60,000 plants and the NARCLIM 2.0 projections. And so for a restoration site, it identifies a species-specific genetic collection areas, and it also identifies future climate matched areas. So let's have a bit more look at that, what that means. So a genetic collection area is an area across which gene flow naturally occurs for a particular species or did occur prior to habitat loss and fragmentation. So here we have outlined in purple is a genetic collection area. Again, it's effectively local provenance, but based on genetic data rather than generalizations for the red cedar. So the pink dots are herbarium records, give you an idea of the distribution of the species. And what this purple genetic collection area tells us is that gene flow for this particular species occurs across a relatively large area. And so if you were to source appropriate local material for this species, you could source it from, that's Brisbane in the north, all the way down to Jervis Bay. Now, as I mentioned, we can't make generalizations and genetic collection areas vary among species. Here's another relatively large genetic collection area. This is for the Port Jackson fig, which extends all the way from Townsville to the southern extent of the species distribution. So, and here's another one, common Wilkia, slightly smaller, extending from, it indicates more restricted gene flow across the area. And then you have something that has an even smaller genetic collection area, the Hardenbergia violacea. So by focusing your seed sourcing on an area where gene flow occurs, you align with natural processes because you're not mixing anything that didn't already share genetic material over time. So the Restore and Renew web tool also gives you future climate matched areas. So these are areas that are, and they're in this orange color. These are areas that are currently experiencing climatic conditions similar to what the target restoration site is projected to experience in the future. So seed sourced from these areas is climate ready because those plants are already experiencing the future climatic conditions of the restoration site. So here we have the Port Jackson fig genetic collection area, and the areas in orange match those of the future climate conditions that are projected for the restoration site. I just picked a few sites in the area and to go show you an idea of how much it can vary, but you can use the web tool. This is actually site specific. So I just put in, this is the Bulwara restoration site. You can see that the climate that is currently, so the climate at the restoration site in 2070 is projected to be what is currently being experienced to the northwest. And then if you look at this was, I think we had someone from this Landcare group. This is on the coast near Friends of Tomarine National Park. I think there was someone here. So the conditions at that site are going to be projected to be similar to what is currently being experienced on the north coast. Whereas if we look at a restoration site west of Nabiac, Walamba Valley, is projected to experience conditions that are similar to what's currently being experienced in the Hunter Valley. So what you can do is you can use the Restore and Renew web tool to design genetically diverse climate-ready restoration by considering both the genetic collection area and the future climate. So to maximise genetic diversity, you want to source seed or other plant material from multiple plants at multiple sites across the entire genetic collection area. So you can, the schematic representation of a collection strategy here, each of those black things as a seed packet. You can imagine you want to combine, you want to incorporate in your restoration site material sourced from multiple sites across that genetic collection area. And you can further support the climate resilience of your population by including seeds sourced from these future climate match sites. For example, if you bring in some material from these areas that are mapped as experiencing conditions like what your restoration site is projected to experience in the future. So there's all these wonderfully worthwhile things you can do using Restore and Renew, and I thought it's worth quickly highlighting some of these, particularly relevant to those of you working in highly fragmented, heavily cleared areas. You can actually recover genetic diversity and reinstate gene flow disrupted by habitat loss and fragmentation. We can move beyond outdated generalizations regarding local provenance, which are going to lead to low levels of genetic diversity, and you can source seed from genetic collection areas that align with natural gene flow. And we can enhance climate readiness by sourcing material from areas already experiencing climatic conditions similar to the projected future conditions of your restoration site. And importantly, we can avoid wasting time and money on failed restoration by shifting the focus from the quantity, in other words, the number of plants that are going into the ground, to quality. So we're shifting the focus from plants in the ground to restoring populations. And also we can use the web tool to minimize inbreeding and enhance reproductive fitness. We can also use the web tool to maximize adaptive potential and increase the chance that restored vegetation can adapt to diseases and changing environmental conditions. And we can overcome the limits to seed availability. And this is something I haven't mentioned yet, but it's very important, is that given that the availability of seed or plants is a huge constraint in restoration, is we can overcome limits to seed availability by discovering that for many species, the area from which seed can be safely sourced is actually much larger than expected or anticipated. Now, I won't spend too much time on this slide, but species available on the web tool so far, there's 100. We have, hopefully in the future, there'll be many hundred. So there's a whole heap of other species that are close to being added to the web tool. I wanted to include this slide just to talk a little bit about the back end and what the data looks like. So these are screenshots from our genetic database, and these are species that are not yet on the web tool, but they're close. So the grey dots represent herbarium records, the distribution of the species. The purple dots are a site where we've gone, or one of our collaborators, and they've collected a leaf sample from 6 individuals. It's a site. It's a collection of leaf material for genetic analysis from 6 individuals at a site. And so those that are purple have already been sequenced, and those that are orange have been collected but not yet been sequenced. So you can see for forest red gum, we're getting pretty close to finishing that species off. Whereas grey box, we need to do some additional collections out in the west of the species distribution. And Kurrajong, again, we've collected lots of material, but it's not yet been sent off for sequencing because we still have a little bit of additional collection to do out further in the west. Now, before we jump into using the web tool, I just wanted to briefly highlight there's 2 ways you can further maximize the success of or the climate readiness of your restoration plantings. Now, an important one is to not pull provenances and maintain maternal lines. So seed collections can be better used in restoration if we keep the seed from different sites and ideally different mother plants separate when collecting, storing and propagating. So here's a research paper here which gives at least 10 reasons why it's a good idea to maintain maternal lines. But briefly to summarize why, so all the seed from one individual belong to the same maternal lines. They're siblings, they're closely related. We want to keep them separate or be able to identify them so that when we plant them out, we mix them all up. So we're not just planting siblings all next to each other. So we want to encourage outcrossing by planting those maternal lines across a site. So you're not putting siblings together. And importantly, we also want to be able to isolate hybrid, inbred or planted maternal lines. You can imagine if you have material from, actually, no, I'll use this example. So here's a study from the Hunter here. We have river red gum. We had 10 mother trees along Wollombi Brook. We sequenced the mothers and the offspring, or the seed and seedlings. And what we actually found is that, so here's the Wollombi Brook individuals. And what we've subsequently found is that one of the individuals that we sampled looked natural. It looked like a natural population, but we subsequently discovered, based on its genetics, that it had been planted. It aligned with material that was from the southwest. And because that stand was so inbred, they were all trees planted, presumably collected from one plant, closely related. All the seed produced by that individual was hybrid with cabbage gum, Eucalyptus amplifolia. So because those maternal lines were maintained, we were able to pull those hybrid individuals out of the collection. Whereas you can imagine, if you're putting all your seed from multiple mothers in the one bag and you discover there's hybrids, it's impossible to pull them out again subsequently without genetic study. So another point I wanted to highlight, and this may be obvious to many of you, but worth highlighting, is that don't restrict yourself to sourcing seed from only the target plant community type. So if you're restoring dry rainforest, you don't need to restrict yourself from sourcing seed only from dry rainforest. So while plant community type is a really convenient way to partition floristic diversity, it doesn't actually reflect the evolutionary history or adaptive potential of a single species. And if you restrict yourself to resourcing material only from that plant community type, you're going to end up with a really low genetic diversity and a low adaptive potential. So in summary, to maximize the success and climate readiness of your plantings, you want to remember that you're restoring self-sustaining populations and need to sensibly maximize genetic diversity. And you can use the Restore and Renew web tool to do this. You want to grow your plants and seed, or collect your seed from multiple plants across multiple sites across the genetic collection area, including sites that match the predicted future climatic conditions. And you want to keep seed from different sites and ideally different plants separate during propagation so that you can mix them together at planting to maximize outcrossing. And don't restrict yourself to sourcing seed only from the same plant community type. Before we jump into the demo, just want to highlight my email address was on the first slide. Feel free to contact me after this and stay in touch by subscribing to our email newsletter, practitioner updates from Risa. You can subscribe at that address there. We also produce an annual publications booklet, which brings together the many publications that the team produces. You can read abstracts in that single booklet, and also we have a number of stories there published which are relevant to restoration. So we're going to jump into using the web tool now. Okay, how do you select additional PCTs to incorporate into seed collection programs for restoration of fragmented ecosystems? So I guess although PCTs or plant community types are a really convenient way for us to talk about ecosystems, what we're suggesting, or what needs to happen, is that you focus instead on plant species. They're the, I guess, the building blocks. And so you kind of, you may be looking at restoring floodplain dry rainforest, but to do that, you're looking at the individual species and you're sourcing those seed for each species, and it can be quite different. You're kind of ignoring the fact that that's a particular ecosystem you're trying to restore. And I can come back to this question after if you feel like I haven't answered that, Isabel. Yeah, so, and thanks, Tash. Are there ways volunteers can get involved with the collection side of Restore and Renew? That's something I want to do. So it's something we tried to do about 10 years ago when we thought of Restore and Renew. The collection of samples is more expensive than the DNA sequencing for us in terms of getting people out there to do the collections. We have some workshops coming up in Sydney on the North Coast shortly. I should have actually included that slide in my presentation. If you go to, actually, I'll just include, in a moment, I'll put some links into some workshop that's coming up in Sydney, and in the afternoon session we're actually training people on how to use our collection app. And yes, we would love to I'm particularly interested in getting Landcare groups to help. There's constraints with licensing in terms of where the collections happen, but hopefully yes, we are definitely interested. And there's another question here. Is there any research into Aboriginal cultural practices such as trading and planting culturally significant species like Brachychaeton and Poplanaeus on genetic diversity? Yes. So we have someone in our group called Monica Fay, and so her research area is biocultural restoration. So it's the co-restoration of a species along with the cultural practices. So that's something that we're very interested in looking at. So we've got some research. I can put, I'll put some links in the chat once the meeting's finished, and they'll be there. So we've done some work on black bean and also bunya pine. The black bean is particularly interesting because we saw that the genetic relationships across the landscape matched the song lines. So you could see how that material had been moved. So yes, we're very interested in that area. And I think that's the questions for now. And then I'll come back to more after a demonstration. So when you click on the link, here's the Restore and Renew web tool. Oh, there's a question. Meg, did you want that now, to answer that now or later?
Just quickly, Tricia, look, this is fabulous. I just wanted to ask a question. Like, I know you're mapping like the natural gene flow of those plants. I'm just wondering like the impact of nurseries coming in, people putting in different nurseries, and then people selecting plants that weren't sort of meant to be in that area, and the impact of pests like pigs or dogs or foxes or other pest management or pest animals that are going to be sort of spreading plants that, you know, wouldn't have spread pre-colonialisation.
Yeah. So I guess what we're mapping is natural gene flow on an evolutionary scale prior to habitat loss, fragmentation, and disturbance. So I guess what we're showing is that if you want to source your seeds in a way that aligns with natural processes, this is the area across which you could do that. And yes, there's a whole heap of other factors which are influencing current gene flow. I'll give an example later on. There's a grass species, Microlana. You can see it's been moved across the landscape, and we've lost that kind of evolutionary signature because it's just human mediated disturbance. For most species, we can still see what would have, but we definitely notice that human movement. And yes, there's lots of issues like particularly nukes, hybridization, because we're seeing isolation and fragmentation. Nukes don't like to self, so instead of self, they hybridize. And what we're seeing is in recent years, we're seeing gene flow that does not align with what happened on an evolutionary scale. But if we understand what happened on an evolutionary scale, then we can try and match that. Does that answer that?
Yeah, pretty much. Thanks, Tricia.
No worries. So what I'm going to do is go in. I'll show you. This is the web tool landing page on the Adapt New South Wales, and you are looking at a page on Adapt New South Wales website at the moment, are you? Yep. Okay. So I'll just briefly introduce you to what's here so you can come back and look at it in your own time. And as I mentioned, it's easily found by looking at resources, Restore and Renew, amongst all the other lovely resources there. There's useful information like a video summarizing what it's about, what the tool does, who's it for, why use it. There's guidance and resources, case studies, including one from the Hunter here. We have the Eucalyptus camaleudensis restoration project. There's also guidance and resources, which gives you more detailed information on some of the genetics background that I've been talking about today. There's also a frequently asked question and a glossary, or a restoration genomics glossary you can look at there. But we're going to jump straight into launching the web tool. And so there's just 3 steps when you use the web tool. You identify your restoration site. So the current version of the web tool is based on selecting sites. Future versions you'll be able to identify geographic regions. So for example, if you're working in the Hunter region, you can click on that cache. But for now, it's based on sites. You click on your restoration site, you select your species that you want to restore or create populations for, and then you view your seed sourcing guidance. So let's get started. I wanted to put in a site here for the Friends of Tomaree. I think I wanted to go up to Fingal Bay, and I wanted to click on. So you can find sites either by dropping a pin on the map, zooming into it, or you can actually type in search for a location. I've just zoomed to this here. So I know someone in your pre-workshop mentioned that you're interested in restoring coastal dune, high dune, and coastal forests. So let's look at a site beyond Zenith Beach. So the first step is it brings up the future climate. So this is an area where the future climate is actually very similar to what's currently being experienced by the site. And but there's a bit of a shift to the north as well. But that information is not very useful on its own. The next step is to select the species. Now, what I want to highlight here, a really important point, is that the web tool brings up a super long, intentionally long list of species that you can select from. So these species, so depending on the objectives of your restoration project, whether or not you're restoring a particular plant community type. So the decisions regarding what species you're including, you decide which species to select. So this list, this list brings up all the species whose extent of occurrence overlaps with that restoration site. So it brings up species that'll not be, they wouldn't be suitable for your particular target restoration site. You actually go through and select from species, from that list, the ones that you're interested in planting. So, you know, you may be interested in Acacia longifolia. Let's select that. And so it's loading the genetic collection area. The first time this pops up, it gives you a bit of a how to use this information, which is what I've been talking to you about in terms of what a genetic collection area is and how you interpret it. So you close that. And so what this does is, here's the genetic collection area for Acacia longifolia for that particular restoration site. So you can see this species has a distribution that's quite wide, particularly in the south. But if you wanted to source seed and maximize genetic diversity for Acacia longifolia for this site at Nelson Bay, you can source your material across this geographic area. Now, we also, for example, may look at Acacia swaviolens. Actually, this is a really awful example, this one. So this is a species. So what it does, it gives you, if you drop down this arrow, it gives you a little bit more information on what the data shows. So this is one where elevation had a really interesting impact on relationship among populations. So the genetic collection area is quite odd looking. And so if you wanted to source seed for Acacia swaviolens for this particular restoration site, the geographic area over which you would source material is actually quite narrow. So and realistically, given that these areas here are likely don't have the species, you'd probably be looking at sourcing fewer individuals across more sites to try and maximize that genetic diversity in this small area. So it's really species by species. Let's put, okay, so we may have Acacia terminalis. Now what you can do, I'm going to just select a few species because you can select species as you go. And at the end, we'll be able to put in and here's the flannel flower, for example, a relatively small genetic collection area. So if you want to maintain that local adaptation and maximize genetic diversity and not have any unintended consequences of mixing material that was too dissimilar, you would source your material from a relatively small geographic area. So this highlights that for some species, like flannel flower, your Landcare group may look at facilitating local seed collection that suits your needs. Whereas for other species with that really broad genetic collection areas, you may source your material from seed suppliers or other Landcare groups. I'm just going to go down and go for a Floribunda. And this is me just doing a desktop. You know, those people working on this restoration site know its objectives. Actually, I just want to show Angophila floribunda. This is an example where it has a whopping big genetic collection area across the whole of New South Wales. So to maximise genetic diversity for this particular species, you can source plants or packets of seed from multiple nurseries or seed suppliers across the state, whereas others like the flannel flower are more local. Now, another thing is, so when you're deciding what species to include, actually, let's do this one. We'll go with Banksia, the Wallenbanksia. You can, ideally, you would use other resources like trees near me or your local knowledge, but also when you're in the tool, you can see that the pink dots are herbarium records. So you can go, oh, actually, yeah, the Wallenbanksia does occur relatively nearby. Maybe we will include that. And then you can see its genetic collection area. So I'm just going to put in, oh, here's an example of a really tiny one, the Coria alba. It's kind of at the northern end of the species distribution, this site. So it has a really tiny genetic collection area. So another one where you may source your own seed, maintaining maternal lines, and propagate them in your nursery, because any commercial seed that's available for this species is not likely to be suitable for your restoration site if you want to maintain plant in alignment with natural processes. I'm quickly just going to select a few more. Heterotrophola is hard. Actually, this is interesting, and those of you who know more about the area may understand why this is the case. But the Hakea teritifolia and the Hardenbergia have relatively similar genetic collection areas. So if you're collecting those, you could have a collection strategy for your geographic area where you know what species you want to collect from what areas, and you go, okay, I may liaise with X Landcare group to get that material. Okay, Melaleuca quinquenervia, Melaleuca nodosa, and Westringia. So what you can do is you can go through and look at those one by one, but you can also download so you can export GIS files of those genetic collection areas for those species, and you can also download a report. So looking at this restoration site, looking at the species in the web tool so far, there's 13 species that you may be thinking about planting at your restoration site, and you can generate a report, which you can download, which brings it all together. It's a rather, unfortunately, ugly report. It's being a bit slow. I'll just show you what it looks like. So it's a seed sourcing report, brings together all the different species, and so you can see the genetic collection area for each of those. And there's a map at the end that brings them together. So just at a glance, you can kind of see what sort of the variation in the genetic collection areas you're dealing with. What I want to do now is I want to jump onto a different. I won't stop the questions now. I'll do one more example to highlight a few different things. So I'm going to go back and reset location. I'm going to do a site west of Nabiac. So someone registered for the workshop on Thursday, I think, is looking at a site in the Willamia Valley. What I'm going to do is I've clicked on a map, some random site approximately in that area. We're selecting species. Now, what I wanted to do is highlight. Again, I'll quickly go through and I'll just, there was someone who was interested in doing shelter belts in this area. That's what it was. And so what I wanted to highlight is that even something like a shelter belt can be, you can have self-sustaining populations if you design it right. So those shelter belts are going to persist for a very long time, and you can bring together material from quite a large area. So let's put in Angophora costata. Actually, I've clicked on such a random spot that I hope that it kind of matches with what I was planning to do. What I wanted to show is that for these two species, again, a relatively big genetic collection area. So you can source your seed or tube stock from any nursery in New South Wales effectively for these sites. So you want to bring together material from multiple sites to maximise that genetic diversity and avoid that inbreeding. But you've got other species like Corymbia gumminifera, which is much smaller. Again, where you'd be focusing on using your local nurseries and local seed suppliers. Now, Microlina, I spoke about that before. Microlina stipoides, and someone was interested in grasses as well. This is, so for some species, we have this icon that says map data not available. Now, not all species behave themselves in terms of their gene flow across the landscape, and it can't be modelled. It doesn't mean it's not useful information. It just means that we couldn't actually model it accurately. So instead, we have some information on what we know about that species and why. So, for example, you can see this icon which says human impact. Microlina has been planted so widely that we're actually seeing that kind of breakdown in that natural gene flow. Overlapping lineages and polyploidy, which is complicating things. And again, we've got more information. Now, what some species have, actually, I'm going to go to a different species to do this. So kangaroo grass. So here's its genetic collection area, an interesting one, because this is a whopping widespread species, which you could get your material all across Australia. But this shows that for this particular site, west of Nabiac, you'd source your material along the north coast and inland. But when you click on this down arrow, see there, some species have this extra button called related intensives. And if you click on that, it brings up some more information. So we have projects which have been funded by someone else who have more information beyond what is available in the web tool. So this is the Cumberland Plain Knowledge Infrastructure Project. You can download a report, and we know by that button that Demeter or kangaroo grass was in this report. And it can give you some more useful information that can help you. So, for example, here's a static map showing the genetic lineages across the state. So, for example, here you've got that red kind of north coast, then another purple lineage, and then a blue lineage down in the south. But what it does give us is there's this, we call it a genetically optimized seed sourcing strategy. And someone's crunched the numbers to work out how many sites and how many individuals you would need to source seed from to capture maximum genetic diversity, or 90% in this case. So if you want to have a genetically diverse restored population of kangaroo grass that is climate ready, you would source material from at least 6 sites with at least 3 of those located in climate-matched areas. So for each of these species in the intensive, someone's crunched the numbers on how many source sites you need to go to capture that. So it's just worthwhile knowing that that exists. And I'll just go back. So you can see all the intensives in the link on the side. So the Restore and Renew Web Tool intensive projects, there's only 3 so far. Depending on where you are, they may or may not be relevant. For those of you in the Hunter Valley, you can find the full report for the river red gum or Eucalyptus camelliensis. And you can see all the reports have this schematic representation of that genetically optimized seed sourcing strategy. So for Eucalyptus camelliensis in the Hunter, if you wanted to restore a genetically diverse, climate-ready population, you would source material from 4 sites within the Hunter Valley and at least 5 trees within each of those sites. And also you'd source material from the Namoy, Guida or McIntyre catchments to the north. And that is the end of the, I guess, the going through, and we've got time for questions. Actually, I said it would be an hour, and that was spot on an hour. So I guess let me look at the questions. There's a new one coming in. I'm in North Rossby in some case. Wonderful. So thanks, Kel. Yes. It would be good. I will stay in touch in terms of sample collection. And Meg, you've got another question?
Yeah, Tricia, sorry. The mapping, when did you sort of start? I'll just start this video again, sorry. When did you sort of start the genetic mapping of plants? Because I'm just thinking how far back you've gone just to incorporate, the effect that weeds would be having on those genetic populations, because they'll be smothering or impacting native species and also pests moving things about. So what was the sort of starting point with the mapping? Because this is quite new, isn't it?
Well, we're looking at it now, but I don't know how to answer this. But what we're looking at is on an evolutionary scale, because it's the genetics. And yes, I know how to answer this. So when we go through and we collect the samples from across the state, all those dots, so when the data's kind of cleaned, and so there's steps that they use using coding and all these complicated things that I'm not up to date with, but they actually, there's a research paper, I'll include that because it talks about this method. And what they do is they are able to pull out the hybrids. They're able to pull out the misidentifications, which is really common in nukes, and they're able to pull out those that are clearly individuals that are not in the right lineage. So if they've been moved around, so they look at the whole population genetic structure, and they understand what it looks like on a natural evolutionary scale. And for the analysis, or what we share and Restore and Renew, all those unnatural kind of things are pulled out of the data set. So the recommendations in Restore and Renew are only based on, they reflect pre-European disturbance. And if they didn't, like the microlena, we didn't put them up because they'd already been moved around too much. So if you're looking at something that reflects evolutionary pre-colonization gene flow. And so for some of those species, actually for many of them, particularly those with the really large genetic collection areas, we know we had large gene flow across the landscape. That gene flow may no longer be happening because of habitat fragmentation. But we know it happened in the past, so that if we were to mix those individuals, there's no negative consequence because they were already mixing.
Yeah, okay, cool. I just wanted to see how you got to that starting point. Yeah, and it sounds, quite scientific how they sort of, eliminated all that sort of complication of all those other things to get to that sort of pre-colonization blueprint, I suppose you'd call it.
Correct. Yeah.
Yeah. Exactly. Okay. Cool. Thanks, Tricia.
No worries. So we've got another question. So what are the restrictions on seed collection using the tool? Do I need a license? So yes, so our tool is literally just saying, ideally you would source seed from these areas and from multiple sites across this area. And when you're sourcing that material, and licensing is one of the main constraints, something that our group is very passionate about seeing is, for example, national parks opened up for licensed seed collection, because you can imagine that if you're collecting seed from these small and isolated populations, they've already got low genetic diversity. So yes, your licensing is independent to our recommendations. And the simplest, I guess, there's several ways to do this. One, you buy your plants from nurseries and you ask them where they got their plants from. And you, instead of sourcing seed from across that genetic collection area, you're sourcing plants from multiple nurseries. Or you're buying packets of seed and the seed collectors have been licensed and you're giving that seed to your nursery and your nursery is growing that seed. Or you're collecting seed from your local area using a scientific license or on private property where you don't need licensing, or with someone from local land services or from DQ who has a license. So yeah, unfortunately, well, not unfortunately, I guess it's important for these things to be licensed. This tool doesn't negate the need. This tool says ideally this is where you get your seed from, but doesn't say that you can just go and pick it. I hope that answers that, Kate. And Isabel, with species such as Microlina, what's the advice for restoration? Interesting question. Should groups simply collect and propagate from their own site, or does it not matter because the genetics are so modified anyway? So yeah, there's very few examples of where this is happening, but for those, yeah, it's kind of, you still want to maximize genetic diversity because if you collect only from locally, even though they've been moved across the landscape, you're likely to encourage inbreeding. So you would still source material from multiple sites and multiple individuals, but you would be less concerned about sticking within a particular geographic area with something like Microlina, just because it's already, what's that saying? The horse has already bolted. We've already moved it around too much. So another question from Tony, applying Restore and Renew guidelines, what will the native nurseries of the future look like? Yeah, so I guess this all comes down to having seed collectors or regional seed banks, and nurseries that have a really good understanding of where their seed comes from and they're maintaining maternal lines. To give you an example, we recently, with my Broke-Bulga Landcare hat on, we had a community field day for River Red Gum. And so Trees and Newcastle Nursery did a great job of maintaining maternal lines in the nursery. And so when the plants came out in the field, we were able to give everyone a selection of plants from different maternal lines so that when they planted them, they could mix them all up so we weren't encouraging inbreeding. So nurseries in the future are going to keep their maternal lines separate and they're going to understand where their seed comes from, and they're going to be able to get a phone call from you and you're going to say, I want X species from as many sites in this area. What have you got? And they're going to be really easily able to tell you what they have. So yeah, good question. And it's going to be an exciting space. So what's the best way to access seeds? Really good question, Yuli. There's commercial seed suppliers. There's multiple. There's also inspiring projects like the North Coast Regional Seed Bank. On the North Coast, the various Landcare groups are involved in that, and they're setting up a system. I think it's super inspiring and worth looking at for inspiration, where the Landcare groups and the community are getting involved in the seed collection. And if you deposit seed into the regional seed bank, you can withdraw seed from the regional seed bank, and where all the seed are being collected from is being tracked. And also, yeah, commercial seed suppliers, there's several. I wanted to say something else with that question and I've forgotten what it was. Oh, that's what I wanted to talk about. So, for example, back to Eucalyptus camaleudensis or river red gum, Katie Elsley from DQ, Saving Our Species program, they're setting up a regional seed bank in collaboration with local land services for that particular species, because you don't want all these multiple landcare groups sourcing their own seed when we could all be using the same source populations or various combinations of. So if you have a regional seed bank, then you can request the seed or request the plants from that. Liz, identified birds. Yeah, I've only read half that question, but I'll highlight an important point. What we've found is with the genetics is we ignore subspecies delineation, because so if we're working on a particular species, we collect all subspecies, and we found that in many cases, the subspecies delineation based on morphology doesn't align with evolutionary relationships. That's just an aside, but I'll just jump to the second half of that question. Yeah, so I guess so this question is saying that, yes, we have different subspecies and we can see morphological differences in different species. Many of these, plants have a lot of polyploidy levels where they duplicate chromosome numbers, etc. And Lamandra longifolia, I think that's one that's going to be like Microlina. I don't know how. Actually, I'm curious enough to have a look at our database whilst we're talking. These are all species. Lamandra, I imagine, has been moved around the landscape. Sorry. So yeah, if we don't have a species in the web tool, you resort to general precautionary simplified local provenance guidelines, which we know are not ideal and we know are not going to create viable, resilient populations. Actually, Lamandra longifolia, interestingly, I might even share my screen so that you can understand a bit more about what it all looks like. So this is just our database that looks at what we've collected. Again, the purple dots, we've sent the tissues off for sequencing. So I'm not quite sure why Lamandra, actually, this is interesting. So Lamandra longifolia, we have sequenced material from across the whole state, really good spread, but it's not on the web tool. I imagine it's not behaving and we haven't been able to model it. And it's one that's kind of sitting in the too hard basket, which again doesn't help. All it really means is that just try and source your material from multiple nurseries or multiple sites. So another question. And nurseries have required record or where the seeds are. So this is a question from Dave talking about sourcing from nurseries, and are there specific nurseries that have the required record of where the seeds were originally sourced from? So ideally all would track, but the degree of, someone may say I collected from Singleton, or someone else may say a specific location. So they all should track where their seed comes from. And I guess my best suggestion is to get to know several nurseries, and you'll find one that you think is doing a better job than others, or one that you prefer. So to give you an example, jumping back to seed, So Yass Area Network of Landcare, Climate Ready Re-Veg Project, they source their seed from multiple nurseries, or multiple seed suppliers, and then we've actually gone in and genotyped the seed that they sourced. What we've found is that even the seed from really good suppliers is heavily inbred, it's misidentified, it's hybrid, but overall, ignoring those problems, when you source material from multiple nurseries or multiple seed suppliers, you do increase genetic diversity, but there's a whole heap of other informations. And thanks, Kel. I'll send you an email after this to stay connected. A question from Isabel. Does polyploidy make species more or less adapted and adaptable, or less? I don't know the answer to that, but I will say that polyploidy complicates things. And you can have polyploid races that don't cross, or they may look identical, but they can't mate, and it's probably not something I'm up to date on. So it's a yes or a no is the answer. So I think that's the end of the questions. Is there anyone else with questions? Yes, it's a hopefully in the future we have this web tool that has many hundreds of species in it, and you can but for now, even when you look at our area, most of your sites, you should have a good handful of species that you can really, so instead of having a restoration site where you go, I'm going to plant 70 species and you just plant, you focus on the number of plants you're putting in the ground, you may have 5 or 6 pet species that you really focus on making sure that you're capturing that genetic diversity and adaptive capacity for those target 5 species. So Joe's got a question.
Yeah. Hi, Tricia. I've just a quick question just about the rate at which you're uploading species. How, like, have you kind of got an average number that you're doing per year? Like, if I was just thinking, if we're just looking at doing some mapping for one of our Landcare sites for planting, but if we're doing that now, is it worthwhile us coming back like in a year's time and reassessing those species? And once you've entered the data for a set species, will you revisit that and update data, or once it's entered, that's kind of it and set and you're moving on to more species? So I'm just wanting to know if data that's in there now for certain species, will that change over time within the near future, and how many species are you uploading per year, say?
So currently, and again, we are listing the new species in our quarterly newsletter that we send out. On average, there's only about 6 or so, a handful of species every 3 months. Restore and Renew has been largely unfunded ad hoc money from here or there until, so Adapt New South Wales or DQ gave us support to update the user interface. But in terms of the species on the web tool, it's been very ad hoc, and we're really hoping for support to fund us putting more species on the web tool. It's not currently funded. So I imagine in the near future, we're going to be adding 6 or so species a quarter. But hopefully, ideally, we will get larger, more substantial support so we can add hundreds of species each year. The infrastructure's there. We could literally put thousands of species on the web tool within 12 months if the resources were there. And in terms of so yes, if you're working on a restoration site, definitely like check in every 3 or so months and go, oh, they've just added. So for example, Eucalyptus triticornis, forest red gum, very close to being uploaded. It'll be there soon. It just hasn't been uploaded yet, and it would be relevant to many restoration sites. And yes, species do get updated. So it's worthwhile if you're, as part of your annual review or something, just check out what's happening, particularly those that have those really narrow genetic collection areas. A really interesting space is currently, our analysis is all based on a DNA sequencing technique called DARTS, or it just looks at a subset of the genome, but it actually is representative. In the future, like in the next few years, we're going to be using whole genome sequencing, so which means that we'll be able to look at traits and look at more information and also update the analysis just by adding in. Currently, we can only update in batches of 96 individuals we send off for sequencing. When we move to whole genome sequencing, we'll be able to send off individual by individual. So we'll be able to more quickly update species. So yeah, it's worth looking back. And if you're interested in a particular species, so our funding model, so we do this kind of work with threatened species through the New South Wales Saving Our program. We've supported that program. Funding is kind of species by species. That's not something we want to do with Restore and Renew. Ideally, we would have, funding for a handful of species like we did for the Cumberland Plain Knowledge Infrastructure project I showed you. But, if you're putting in a funding application for a project and there's one particular species, your key, one of your favorite species that's not on the web tool, think about putting in funding for that particular species into your funding application, or think about asking for philanthropists, or just looking for ways that you can do this kind of work or fund it. So yeah, you can chat to me about that. Hope that answers, Jo.
Yeah, it does. Can I ask, might be a bit bold, is there like a rough costing for how much it does for like one species, or is that kind of...
Yeah, it's really hard, and my manager would hate me answering this question now, because I guess what we prefer is those multi-species regional projects. So, for example, ideally what we would love is, looking at the Hunter Valley, for example, a project that looked at a dozen species across the Hunter Valley, and it would be a couple of 100,000, and I'm rounding into, whereas single species is tens of thousands. And we're at the point now where we want to stop kind of doing single species, when we want to do geographic areas. But, if there's an interesting project on a species that we're already working on, it's just a matter of if there's some research interest in that as well, then that we can do the species by species. But I'm definitely happy to chat. And then I'm not allowed to talk numbers, but I got to kind of pitch the projects to the managers and say, okay, there's this group doing this, and what if they included this in their funding application? How much would that cost? And again, it's really good for us if Landcare groups can facilitate collections, which is where it's quite expensive for us.
Yeah, I just adding to that with the collections, I know there was a few queries about it, but we do, like I'm also involved on the Tomaree Peninsula with the Friends of Tomaree down there. And we do, I'm sure there's a lot of other Landcare groups that we have a license to collect and do our own seed collecting.
Perfect.
So this has been really interesting for me.
Because there's a few species there that you probably would need to rely on doing your own local collections. Yes, and that's where you do things like maintaining those maternal lines. Even if you're getting a little individual flatten flower, you keep the seed from that individual in a separate bag to this one.
Yeah,
okay. And that's where it comes down to nursery practices. A lot of nurseries balk at that kind of maintaining maternal lines, but really it's just they're different accessions, and it's just a matter of tracking them like they're, instead of a pooled sample, they're just an individual mother. But that's great. So the Tomaree Peninsula group, given that they are, have a relationship with national parks.
We do. We have a license to collect in the park. So we do. Yeah, we collect. We have a busy collecting period over, any period, but over the 6 months we're seed collecting.
And where are they propagated? Are they in the
we have the community nursery, which is in Nelson Bay, Nyaga. So that's a council-owned facility. So all of our seed is sourced locally. But it's also interesting. I'll have to look into more about with the labelling of seed collections as well, and maybe provide a bit more detail on our collections. But I was just saying that there is probably, maybe throughout the Hunter, I'm not sure about licenses with other Landcare groups, but there's probably quite a lot of potential for Landcare groups to help support with the collection of material, which Landcare can possibly, Hunter Landcare can possibly help facilitate that as well.
We have some workshops coming up in Sydney where we have the afternoon session of the workshop is on how to use our collection app. And, we're running these workshops in collaboration with various groups. If this is something that, Hunter Region Landcare thought that they wanted, we could look at whether or not we do this workshop in the Hunter as well, or come down to the Sydney one. And there's a question there, Kate, what was the process for Tomaree? Oh, that's not for me. I guess that's a Jo question.
I can answer that. So Kate, we have the Tomaree, Friends of Tomaree are a Landcare group that Hunter are registered with Hunter Landcare as well. And we have a really close relationship with the national parks that manages the land that we work in. We only work in national parks. So our licensing was done through them. So our license is restricted just to the national park as well. And we report directly to them too for our collecting.
And whilst Joe was just talking, I just put in answer to the question about cultural practices. I just put in a link to an article by Monica Fay on that summarises the bunya pine and black pine work. So that's there if you're interested. For those of you who like heavy science, here's a paper that talks about the actual the process of kind of that talks a bit about the cleaning that happened that I spoke about with the taking out of the misidentifications and the human mediated, etc. Actually, we should be fairly user-friendly. It's in Restoration Ecology, but that's there. And if anyone else has any other questions, they can always just get in touch.
I think that's it. So thank you. We'll finish up. Thank you, Tricia, so much for your time. And it's been a really valuable webinar. I found it really useful, and I've had lots of questions answered and lots of new things to think about, especially regarding PCTs on the Tomaree Peninsula and our seed collecting. That's kind of throwing a bit of a curveball my way regarding seed collecting, which is good. But yeah, thanks so much for your time. If anyone has any more questions down the track, can they contact you directly via email? If that's okay?
I'll send through a PDF of my presentation, which has my email address on it. And I'm very happy. And I'm also particularly enthusiastic about having collaborations in the Hunter, given that I live here in the Hunter.
Yes,
so I'm very happy to chat and very happy to talk, Joe, more about your seed collection strategies, etc.
Yeah, thank you. So Helen, we will have this as a recording, and Helen, once at a later date, once it's been edited and cleaned, she'll be emailing that out to everybody, so you can rewatch it and pass it around. So thank you, Tricia.
Thank you. Chat to you later. Bye.
Thanks.
Thanks, everyone. Bye.